Welcome to Friday afternoon, and welcome to some really great speakers this afternoon to kick off a discussion around the transforming regulatory pathways, CEiRSI, particularly as it relates to the promise and requirements of model-informed computational evidence. All of our speakers this afternoon, bar myself and Alex, are from MHRA. I should introduce myself, shouldn't I? I'm Phil Brown. I'm Director of Regulatory and Compliance at the Association of British Health Tech Industries, and it's great to see you all. So Daniel is the Deputy Director of Clinical Investigations at MHRA, is that right? Or have I given you a bit of a promotion?
Yeah, you have, but that's nice though. Thank you. No, I am the
Well,
deputy head of
it's
clinical
Friday. Have a
investigations.
nice present for you.
Yeah, thank you. It's boosted me up. No, thank you, Phil. I'm the deputy head of clinical investigations here at the MHRA, standing in for Mark Grumbridge, who unfortunately couldn't be with us here today, who's the head of clinical investigations. But I am happy to stand in instead and give an overview of clinical investigations, and I suppose at the end prompt a little bit of discussion about where In-Silico fits into clinical investigations. So firstly, I'd just like to give an overview of what clinical investigations are, I guess, and the process that runs. Obviously, clinical investigations are studies of medical devices for conformity assessment purposes generally, developing new medical devices, something along those lines. Obviously, medical devices have to go through a fairly rigorous, generally, development process because obviously they have risk attached, like all things in medicine. And so that can really mean a rather extended process for developing. It depends obviously on the risk classes and the sort of types of devices that you're encountering. But certainly for things like active implantables or implantable devices, that can mean a developmental time of upwards of 5 years, upwards of 10 years sometimes for implants and things like that. And that can be quite a burden sometimes to manufacturers, a barrier to entry, I suppose, for developing and marketing new medical devices and technological advancements. So that sets the scene, I suppose. MHRA have to review clinical investigations. So once once the device is ready to be tested in humans, an application must be submitted to MHRA, and MHRA must give a notice of no objection before the study is allowed to go ahead. And so the MHRA reviews, well, along with its partner regulators, the HRA, who looks into ethics of the studies and is responsible for the research ethics committees. And we must give a notice of no objection before studies are able to proceed. So that sets the scene, I believe. So these applications have to come to MHRA when a device is not CE marked, or it's CE marked device being used outside of its intended use, and you're developing this for a new intended use. We have flowcharts that describe certain studies and when they are required to be submit an application to us. And so these can be found on MHRA's guidance pages. So the first step in a clinical investigation is that an entire file of the sort of documentation that needs to be supplied to the MHRA. And we have a giant list of this guidance, which we can supply as well, or what we actually need to review a clinical investigation, as well as what the regulations say must be supplied. This must be supplied online through the Integrated Research Application System, or IRAS. Once you've submitted your application there with all the relevant documents, it will come to the MHRA. And of course, these can be anywhere from something like 20 to 30 documents until we had one this year, which was upwards of 500 documents. And they're all quite in depth, quite a lot of science and research and detail goes into it. So given the time and the complexity of these applications, MHRA allow 5 working days to validate that application. And that will basically be an initial sort of review to make sure that the regulations are complied with in terms of objectives and endpoints and all the various things that the current regulations require around clinical investigations. And also that we have actually everything that we need to be able to review the study. And that includes things like varying on and depending on the type of the device, but things like software. We need a full software development plan and documentation set to the standard 62304. I think it's IEC 62304. We need all of that to make sure that the software has appropriate safeguards in place and risk management in place if it's going to be driving or controlling or, involved in this medical device in some way. Sterilization, we need a range of sterilization reports and sterilization summary report to show that this particular medical device is sterilized and it's not going to have risk of infection if it's implanted in the body, et cetera, et cetera. I think I might come on to this again a little bit later, each of the sort of categories that we review, but that initial review allows us 5 days to make sure we have everything we need to be able to actually review and confirm the safety of this sort of device. If we do have everything we need, we will alert the sponsor or the manufacturer or the contact person that we are able to proceed, that the application will be valid, and we'll move on to a 60-day review period. Common types of devices under investigation. At the moment, we see quite a large range, actually. There's definitely some trends. The first one, of course, is software. We're seeing a larger and larger range of software come through for all manner and name of purpose. And so this is a common trend that we are seeing at the moment. Second one, I believe this refers to TAVI heart valves. We do see clinical investigations come through for heart valves and other sorts of implantables and catheters and things along this line. And actually understand that this is one of the sort of areas, active implantables generally, where there is the real promise or potential of benefits for In-Silico. And the third one is brain-computer interfaces is a real booming area of clinical study at the moment, medical device development. This is quite a funny graph because the last bar doesn't quite match up with its numbers, but this is just to give an indication of roughly the amount of applications that we see and review each year. Obviously, 2020 was a COVID year, and that was quite a disruptive year for the MHRA and for the world in general. And so we noticed a drop off of applications in that year. But then since then, it has actually been quite consistent in that we're seeing about 80 valid applications per year. And I just want to emphasize that. So these are the ones that go on to clinical investigation review. And we see it 81 in 2021, 83 in 22, and 84 in 23, and 84 again in 2024. So quite consistent actually in the numbers that we're reviewing. So once we have a valid application, this is just roughly how the MHRA processes these applications. The regulatory handlers, members of my team, will be assigned the clinical investigation, and they will have to sign various assessors from various ranges of specialities to ensure that their areas are being reviewed and being determined to be safe. And this is, of course, in a proportionate basis. So something that has no software will not be reviewed for its software safety. Something that is not sterile will not need a sterilization review. Things that are not implantable or in touch with injured skin or patient contacting won't need a biological safety and toxicology review. But the process generally runs that these assessors will be assigned from their various specialties within the agency. And we will go back and forth in terms of rounds of questions until the assessors are satisfied that this is safe. Or in fact, if they're satisfied that it's not safe, in which case we will notify the company in the form of a grounds for non-acceptance that we have concerns with the study, and for our grounds of either public safety or public policy, we currently do not believe that the study should go ahead. And we will have a meeting with the sponsor, the manufacturer, whoever's in charge at that point, and we will discuss these concerns and outline where we think need to be addressed, and why, if we are going to object, why we're going to object, on what basis, and what needs to be addressed for this study to proceed in future. So this just points to, I think, briefly what I mentioned, which we have a range of experts from across our agency review each of these applications. And this includes internal clinical staff. This includes technical staff who are looking at the the specs of the device and its performance. This includes external clinical consultants, which we bring in to help us in specific areas of study, specific medicine areas, so that we have sort of cutting edge experts able to advise us on these various areas of application. Sterilization, biocompatibility, and toxicology. We also have it reviewed for statisticians to make sure that the plan is statistically sound and is going to deliver on the performance claims that are attempting to be demonstrated by the sponsor or the manufacturer, and also for electrical safety to make sure there's no electrical risks or shock risks, indeed shortage risks, things like that, and software of course as well, to make sure that the software has been designed in accordance with established standards for medical devices, so that you know, some risks are, for instance, that if the software were to be driving a key medical device, let's say something being used in the middle of surgery, and it were to encounter a code failure and just shut down, those sorts of risks have to be mitigated away in medical devices, among others, of course, as well. And also one that's not listed here, which is for animal tissues, where animal tissues are used as part of the design of and manufacture of the medical device. We have to review to ensure that those tissues don't create any undue risks in the medical device or for the patient or participant. So as this briefly speaks to our assessment process, and as I described, we have 60 days for Great Britain studies, or 45 or 65 days for Northern Ireland studies, depending on whether clinical experts are used, which we almost always use clinical experts. So it will almost always be 65 days. And in this period, we are to review the documentation and dialogue with the sponsor or manufacturer, whoever's running the study, and arrive at the point where each of our assessors are satisfied that it presents no undue risks in terms of public safety or public policy, and we are able to say that yes, we're happy for this study to go ahead. And so this is the process that we'll go through. As it says here on the slides, that day 1 of the 60 days is the first day after a application is deemed valid. This is a brief diagram, I suppose, of the report that assessors will submit. Our systems automatically fill out some of these areas, so company name, model number, etc., description. But areas of assessment, the assessors will write a report to detail if they think the study should go ahead or if they think it shouldn't go ahead, and the grounds for why this is. And where we have grounds that it shouldn't go ahead, as I said, we will always convey these in grounds for non-acceptance to the sponsor or manufacturer, and we will have those discussions and those meetings and see if it is possible that these are able to be addressed within the time period that we have for review. And if they're not, then unfortunately, it may lead to an objection. But any resubmission after that period of time will, as long as nothing else in the study or device has changed, we'll just need to address those grounds for rejection satisfactorily, and we will be able to approve that study. It's worth noting also here, I think, that our final letters for when we issue a no objection, or actually for the case of Northern Ireland, it's called an authorization, I believe, will detail our decision, but it will also detail if we have any conditions. Under the regulations, we are able to import conditions on approval of a study. And so this allows us to put, say, on higher risk studies, one common condition that we put is we require a mandatory safety and performance report after usually 10%, but sometimes less, or more of the study cohort has been treated or diagnosed or has used the device. And we want this safety and performance report to give us initial information around, say, if it's implantations, the implantation procedure, how say post 30 days, what the safety profile is looking like, any serious adverse events that have arisen in that time, any concerns, any device deficiencies that we've been made aware of. And in that time, the company is not able to continue with that study until we approve those provisional reports. Other conditions we put as well is a common condition that comes up sometimes is where testing for medical devices should be done before it arrives at us, but sometimes there are delays with that, and it might just be an extra week or two before the testing has actually been provided to us. And in that instance, we can sometimes use the conditional no objection to allow a short extra period of time while we await to receive and approve final testing for something like final electrical safety tests coming back, etc. And as I said, the possible outcomes are no objection, where the study is allowed to proceed; conditional no objection, where we apply conditions, and as long as they're met, they need to be met before the study can proceed; or an objection. In the instance of an objection, as I said, we will detail the grounds for objection, and these must be addressed before the study is allowed, once in a resubmission, and then the study will be allowed to proceed. So no objection is just detailed out in the next one, I think, and the one after that is conditional no objection, which I've briefly spoken to. I can give a brief. So a few reasons, common reasons that we might see is that wrong classifications for software, and so wrong documentation to reflect, insufficient clinical design to prove what you're actually trying to prove as a medical device and the claims that you're making. No sterilization validation, no proper biological safety report. The risks don't actually outweigh the benefits, which does come up sometimes. A few others, I think, toxicology testing and biological safety testing, I think I touched on that. Certainly around software, inappropriate classification and inappropriate software risk analysis, and not covering all the risks and mitigating properly all the risks that the device is likely to, that the software is likely to encounter. Once studies are approved, you must submit an amendment to us. If anything in the study actually at all changes, it must be sent to us in the form of an amendment. This includes extensions, changes to the device under investigation, and in some instances, that may warrant a new submission depending on the nature of the changes. Any changes to the protocol, any changes to the study documentation, changes to the investigators, etc. We must also be notified of any deviations that occur during the study. And if a large amount of deviations are coming in, then that might trigger some questions being asked, and we may have to have further dialogue with that particular sponsor or manufacturer as to why the protocol is being deviated from, and why this is occurring regularly, things like that. And we do also have a power under the regulations to withdraw our notice of no objection at any time, actually. So if we have concerns, then we're able to press on potentially with withdrawing the notice of no objection. But preferably, we like to do this in collaboration with the sponsor, as we can address things collaboratively. The combined review process, where both a medical device and a medicine are being reviewed at the MHRA, and both are investigational. We have a combined process for this, where it is submitted through Medicines CEiRSI. Well, first we pre-validate all the documents and it's submitted through Medicines CEiRSI, and we will conduct our reviews in parallel with the clinical trials unit, and we will each separately issue approvals for our aspect, for the device and for the medicine from clinical trials. The IDAP pilot pathway is something that's actually in its final stages at the moment, where the MHRA, working alongside some partners in the health regulation space, are working as to how we can streamline access to medical devices through joining up areas of regulation in the UK to get innovative devices to patients. I won't speak to this too much, actually, because it's in its final phase now. It's in its evaluation phase, but it's interesting to be aware of. And in that, we tested 30-day reviews and reviews in conjunction with all of our with our partners, and we have some learnings which are being evaluated right at this point in time. So some points for discussion with In-Silico and its place in clinical investigations. Well, firstly, the first point to note is, at this particular moment in time, there is no plan to update legislation for clinical investigations. But it may be that following the work of the CEiRSI and the regulatory sciences, it may be that there is opportunity and benefit for both patients and for access to technologies where we might be able to look into some form of updating clinical investigation regulations to further enable this. MHRA has significant technical and clinical capacity to manage an application that has in-silico aspect to it. I understand that in-silico really feeds in, especially one of the strong use cases is around bench testing and early phase testing. And it will be a real pleasure to work alongside the CEiRSI and figure out where and how we are able to best integrate sort of in-silico testing with bench testing and enabling that to bring devices to, well, hopefully enabling devices to come to market faster using these various technologies. We will learn from the pilot, and MHRA will update our guidance as necessary with input from experts. So it will be excellent for us to work alongside the CEiRSI here and see the learnings that come out of it and how indeed we might be able to update our guidance for how clinical investigations are to be run. And also ethical aspects. How will an in-silico study impact participants? And what do they need to know? So some points for discussion.
Thank you so much for that. I mean, there are some really good points for discussion. I just want to remind everybody that the Q&A thing is open, so please jot your questions and answers into there so that we can have a discussion at the end of the session here. But in addition to those, Daniel, I was really intrigued by the comments on risk and how they sort of potentially outweigh benefit, and like when you review trials, it'd be good to sort of see how in-silico fits with that. And of course, I think that there are new versions of IDAP coming through, aren't they? Which I think on your roadmap is around June time, I think, for and it's a new name now, isn't it? Innovative pathways. I can't remember the full name of it now, but it's a new process after the pilot now. So it'd be interesting to see where that goes. So thank you for that. So some additional thoughts maybe for people as we go towards the Q&A. As I say, don't forget the Q&A. We want plenty of questions and interactive stuff now. So thank you, Daniel.
No problem. Thank you.
Andrea, do you have, or Lara, can we get Andrea's slides up now? Perfect.
Yeah, sure.
Fantastic.
Andrea.
Thank you so much, Phil. Thank you. So my name is Andrea Manfrin. I'm the Deputy Director of Clinical Investigations and Trials at the MHRA. So in my presentation, I'm going to cover 3 main points. The first is who we are and what we do. The second is the challenges and opportunities of in-silico trial through the lenses of the clinical trials unit. And the third, I'm going to give you an helicopter view of the new clinical trial regulation that will come into force next year, in 2026, but they will be a game changer and potentially support in-silico trials. So to begin with, this slide is very important because in some ways encapsulating what we do in a nutshell. So the red circle includes all the clinical studies. The blue circle, the interventional studies and some clinical trials, but the green circle is the most important one because it is focusing on the clinical trials of investigational medicinal product. And we at the MHRA, in terms of clinical trial unit, we are really focusing on clinical trials that are involving investigational medicinal product. So these are the trials that are coming to us for review, assessment, and approval. So in the clinical investigation and trials unit, we have 3 teams: the clinical trials team that is looking at drugs, the clinical investigation that is looking at devices, and we learn from Daniel how important it is, and the clinical investigation and trial operation team that is doing all the data analytics, managing the workflow, and also inputting a lot on the new clinical trial guidance as well. So 3 teams with different tasks, but they are all joined very well together. This is a very important slide because in some way is summarizing in a diagram all the process that is involving a submission of a clinical trials. Well, I like to remind you that by clinical trials, we means every single trials that is involved in medication. Medication can be a chemical medication, biological medication, ATMP, advanced therapeutic medicinal product, any type of medication that is bringing innovation or classified as innovative medicinal product will come to MHRA. And this is the process that the submission is going through. It is defined as a combined review because, as you can see from the left-hand side of the screen, you have the submission using the CEiRSI portal, which is owned by HRA. This submission is going in two different directions simultaneously. One submission will go to REC for the ethic review, and the other submission will go to MHRA, so will come to us. We, as at the MHRA, we are defined as a statutory unit because by law we have to do our job within a specific time frame. And the first part is 30 days, and what we call it is the first review. During this first review, a clinical trial could have, generally speaking, two options or two endpoints. The first is acceptance, and the second is what we define as ground for non-acceptance, which means that we are raising question. And this question could be, similar question could be raised by the ethics committee, and they call them RFI, request for information. Once we raise the question, the clinical trial goes back to the sponsor, and then the sponsor has 14 days, unless they ask for an extension, to reply to our questions. After the 14 days, the trial comes back to us with a response from the sponsor, and then we have 16 days to review the answer and accept or reject or apply conditions. All in all, this is a 60-day process that can be extended for specific type of medication, such as ATMP. But this is in some way summarizing the full process from start to end, showing that we are reviewing in parallel with the ethics committee, and we are issuing a final letter of acceptance if the trial is accepted in combination with HRA as well, with the REC committee. So this is just a brief summary of the documentation that usually the sponsor needs to submit to support a clinical trial application. The cover letter, the application form, which is downloaded from the portal, the protocol for the trial, information regarding the summary of product characteristics, the investigational medicinal product dossier, labeling, information regarding the manufacturing authorization, GMP, and if the trial is involving pediatric population, what we call the PIP, Pediatric Investigation Plan. This is pretty much a kind of a standard package with the majority of the information that are needed for each phase of trial, generally speaking. But it's also important to know what the MHRA clinical trial unit does not review or does not assess. Well, the first point is that we are not looking at the participant information consent because it's not our activity. We are not assessing the suitability of the investigator. We are not evaluating the suitability of the trial in the context of the overarching drug development. And more importantly, we are not assessing observational trials. So these are activities that we do not do because we are concentrating on the trial that are included in the green circle that I show you at the beginning. So we have, generally speaking, two types of submission, what we call a CTA, clinical trial authorization, and the second is a clinical trial notification. The main difference is that the clinical trial application for authorization is assessed by our assessor. The clinical trial notification is notified according to specific criteria that are listed on our website. And these criteria, if need be, they will be assessed by low risk assessor. For the clinical trial notification, these specific criteria are including, for time being, right now, only clinical trial in phase 4, some of them, and some phase 3 trial. Meanwhile, for the clinical trial application, all the trial could be involved from phase 1, first in human, phase 2, 3, and 4. So now it's very important to understand who assess what. So within the clinical trial units, we have 3 teams, what we call the non-clinical team or pharmacology and toxicologist, the quality team, pharmaceutical, and the clinical team, medical doctor. So the non-clinical team assess the basic information regarding pharmacokinetics, the evidence and efficacy of the medication in animal models, so what we call preclinical, the safety pharmacology, potential cardiotoxicity, renal toxicity, liver toxicity, or central nervous system toxicity as well. Other type of toxicity, genotoxicity, carcinogenicity, and potential impact on reproduction, particularly if the trial is involving women with childbearing potential. Regarding the quality team, they look at safety in terms of the medication, the route of synthesis, the structure, potential impurity, batch analysis, stability, composition, and potential contamination via viral or non-viral elements. The medical team is looking at dose, the duration, the dose escalation, which is increasing the dose, the potential efficacy of the medication, and overall the balance between the risk and the benefit, because our review is called regulatory review, but is a review focused on safety. The medical team also look at the trial population to ensure that the inclusion criteria are correct and are not posing excessive risk to the population of the participant, including in the trial. The medical team also, they look at the potential hazard, and they are looking also at the risk mitigation that are embedded into the protocol. They do the safety monitoring, and they also assess if the trial design is appropriate and based on available data. So which are the regulatory challenges of In-Silico trial from the perspective of non-clinical team, the quality team, and the clinical team? So for the non-clinical team, the first question is how representative are the In-Silico models? The second is that could be that is oversimplifying the biological system, and are the In-Silico model reliable? For the quality team, one of the most important questions is the investigational medicinal product using In-Silico sufficiently representative of the IMP, or the investigational medicinal product proposed in the trial? How would the validity of the In-Silico method be assessed for the situation if it is intended to model? Meanwhile, for the clinical team, it's very important to understand if the model and simulation can be used as a supportive tool, but not as the main trial. Modeling can be used to optimize the design of the trial and to calculate the dose in subgroup, for instance, in pediatric population, elderly, and impairment, or renal impairment, and for the safety perspective as well. Now I have a couple of examples that are just in some way creating a kind of a real environment. And here the clinical team provided a couple of examples, which is very, very important. So the first one is that the modeling can be used to justify the dose of a medication tested in a clinical trial, including children or adolescents, when the data from adults is available and using methods. But in this case, the model is used to calculate the dose for the pediatric population, but is not intended to replace the actual study testing the safety and the efficacy of the medication. And the second point is the Bayesian modeling can be used to guide dose escalation and decision in early phase studies. However, modeling cannot replace the need of conducting a dose escalation study. So in a nutshell, the clinical team is suggesting that the In-Silico trial can be used as a supportive tool for the clinical trial application, but they will not, at the moment at least, replace a real trial. They will be an add-on. But in the next slides, there are some other very interesting information and consideration from the clinical pharmacology perspective. So, and the assumption is the computer simulation could replace clinical study or trial in the normal clinical pharmacology package. Well, In-Silico trial involves prediction of pharmacokinetics. That means, how the medication is going around the body, and in more complex situations, including physiologically based pharmacokinetic modeling as well. The PBPK can support first-in-human trial, and the clinical pharmacology team is encouraging this approach, and they are suggesting that this is supported also by the publication of the ICH M15 guidance, that is a guidance specific for the development of model-informed drug development. And in this slide, there is a kind of an analysis provided by the clinical pharmacology team regarding the pros and cons of In-Silico trial. So if we look about the pros, of course, the study, less clinical study are needed. So thank you. There is a cost reduction, of course. There are ethical implications, and is potentially very good for difficult population to treat, such as children and pregnant women. And of course, also it could improve the knowledge behind the study, less risk. For example, if the dose is too high, so the In-Silico is safer, and it can provide also mechanistic understanding of the other risk, where mechanistics is represented by the equation that are described to, in some way, show the physiology and the drug perspective and the solubility, for example. In terms of the cons, there are issues regarding the risk due to assumption that the model has some gaps, or there is gaps in the knowledge of the model, technical expertise in modeling the PBPK or the population pharmacokinetics as well. And there are also regulatory questions on the models that needs to be addressed in the future. So in terms of having a regulatory submission, we know that EMA and FDA, they provided guidance, but since the MHRA left the EMA, now we have worked on the generation of the ICH M15 guidance. And as I mentioned before, these are important guidance for model-informed drug development. And we are advising the sponsor to look at the guidance because they provide added value and a lot of information for a robust submission. So the last point that I would like to touch is regarding the regulatory reform of clinical trials. So as you probably are aware, in December 2024, the new clinical trial regulations were laid before Parliament. In February, the new regs were debated in the House of Lords and Commons, and now we are waiting for the final debate in Northern Ireland. But the key themes are the following. The first is that the new clinical trial regulation will ensure patient and patient safety. They will create a proportional and flexible regulatory environment, and they are aiming to cement the UK as the destination for international clinical trial, not just for national clinical trial. And they will provide a framework that is streamlined and agile to respond and support innovation. So which are the changes of the new regulation? Well, probably the biggest change is that in the new regulation, we are embedded a more streamlined approach for the clinical trial, initial and clinical trial amendment. This is very, very important because we are introducing the notification for some clinical trial initial and the notification for some clinical trial amendment. There is also research transparency because the clinical trial registration will be before the start of the recruitment, and the result will have to be published within 12 months of the end of the trial. The good clinical practice will improve the alignment between GCP and ICH. The manufacturing and assembly process will be streamlined in terms of information, and we are removing duplicative reporting in terms of the safety side of the trial. My last slide probably will be the final slides for me. Well, thank you very much for listening, and I'm happy to take any question you might have towards the end of the webinar. Thank you so much.
Andrea, that was absolutely fantastic, and not only from content perspective, but bringing us back onto time. It's absolutely amazing. Thank you so much for that. Again, please provide any of your questions that you have in the Q&A. Andrea, what I'll probably pick up with you in the Q&A is maybe some of the things that sit around medical devices, maybe, because you talked a lot about pharma and how that may work, but also things like we're talking in the regulatory perspective, on a regulatory perspective, again, particularly with medtech, on things like reliance models and how we work with those. Be interesting to sort of pick up on if the FDA or Europe are using In-Silico as part of their basis for regulatory approval, how that might affect what happens in the UK, or what additional work we might need to do from that perspective, or how the work which Alex and his team are embarking on might help assist in some of those reliance models. But we'll leave that for the discussion. But thank you so much for your presentation. I'm going to bring Alex in now because Alex is next on the agenda to talk about strategic imperative for regulatory success. And Alex, please, over to you.
Thanks. Thanks a lot. So what I like to do is remind ourselves why we need to reflect on the regulatory pathways that were discussed before, and then try to perhaps give some examples about how we might, some examples about what we mean by in-silico trials and what we don't mean by them. And so I always like to start by reminding myself that the first rule that medicine has and all clinicians have, and therefore all of those that try to support them in their mission to to support patients is not to harm. It's not to introduce any additional harm to patients. But when you actually look critically to the way that we are currently producing regulatory evidence, both for devices and for drugs, we realize that there is a number of ways to do that with bench, animal, human testing that came very clear through the presentations before, and also that there is fundamental questions around safety and efficacy of those products. And the big question is to what extent those processes really are doing their job at the minute. Now, the various avenues for testing have limitations that we're not going to go into a lot of detail, but from limitations in terms of the conditions of testing and how that relate to a scenario of bench testing, to let alone the waste that is produced with those, animal testing in terms of the difference between a species, and also the implications in terms of animal harm. Human clinical trials, we tend to forget they are also a model. Ultimately, they're a model about what happens in the wider population when we test only on a subset of subjects. And then, of course, many of these things, all of these processes can be done incorrectly, and that's why there is good the importance of guidelines and procedures, and good laboratory practices, good trials practice, etc., to make sure that they are well done. But despite all of these efforts, we still have considerable challenges in the fact that still there are devices and drugs that produce considerable harm, despite all of the efforts so far. So we need to reflect about that. And I show usually one example that is a particular device that was taken out of the market a year ago. But that reflects a wider concern and a wider impact in the sector. So it's not just a particular company or a particular device, but more broadly, the number of recalls that we're having pretty much across a lot of the companies of the sector. The other thing that it was really impacted me a lot a few weeks ago is when I came across to these slides from Tina Morrison and Mark Palmer, where they show that actually a large proportion of the devices that actually go through the whole testing process remain and will remain investigational, and there is a substantial amount of patients that go through trials. In the UK, it's about 1 million people a year that participate in one way or another in clinical studies. There is, in the case of these particular studies from the US, there was about 41% of the patients that participating in those investigational studies that actually the products that were used with them will never become commercial. And this is even worse in the case of pharma, which is 5 compounds of every 100, and it's about 68% of the patients that will be used where drugs will be tested that will remain investigational. And that also has different stages or different impact in different stages of different levels of testing of those products. The other thing is it takes a long time, which effectively means that from a commercial point of view is also challenging. And also we have a substantial waste in the amount of research we need to do to actually get a particular product all the way to market. So a complex therapeutic device is on the order of 54 million with a large margin, but actually that is if you get it right first time. Normally there is a large attrition rate, as we discussed before, and that means that there is a huge amount of cost for that is added to that successful device to actually compensate for all the failures we had along the way. And this, in one way or another, ends up being rolled out into the taxpayers. It ends up rolled out in the national healthcare system. So it contributes to the unsustainability of the healthcare in itself. And this again, calls for an ethical imperative to rethink how we do things. And then finally, I want to emphasize that as well, most of these studies that we consider the holy grail, randomized controlled trials, when you really look in detail into those, there is all kind of, inefficiencies in the way that they're run in the sense of that we lose patients and the cost this brings about, but also in terms of the amount of representation that is really captured through those studies. And, there is a number of studies that show that particular demographics actually have are critically underrepresented in a number of the studies, and that many of the patients that participate also drop from those studies as the studies progress, which actually renders some of them perhaps not to have the originally planned statistical power, or simply that they have to be sort of aborted in the process. And this again has impacts not only in terms of cost for the industry, but all of the national healthcare system that is contributing and invested in delivering those trials. So, if you have discussions with colleagues from the NIHR, one of the things that would be very keen is, can we have ways in which we could identify those trials that are likely to be most successful, so that we can actually, invest the energy in those and actually support the generation of good quality evidence. Of course, all of these factors are not necessarily the same across all of the different types of therapeutic areas. There are certain ones that actually have much larger costs than others, and also the size of the trials is fundamentally affecting the cost of those trials as well. But usually there where we want to do very large trials is where the cost is large. And in cases where we have relatively small trials, this is sometimes because of limitations in the design of the trials, or the ability to recruit sufficient patients and follow them up in a given time frame. So all in all, what all of these factors are is that those ecosystems that are better prepared to actually support industry in delivering the best studies will both produce the best regulatory evidence for their demographics, but also be most attractive for innovative companies to land there and provide even more investment. So both from a productivity point of view and from a patient outcome point of view, there is this strategic imperative that I was talking about. Now, there are other sectors that understood this already some time ago. Aerospace is one of them. Automotive is another one. And what in those sectors, the approach is not so much design and test or build and test approach, but actually to simulate or optimize, modify early on, and get your design better improved, and then do the evaluation almost as a confirmation of your design. But by the time you design, you have tools and means that you are fairly confident that what you have designed is of good quality. So what we are suggesting is perhaps these first do not harm needs to translate into simulation first. We are not suggesting that we shouldn't be relying to some extent in some of the traditional routes, but I think the other ethical imperative and a strategic imperative is, if you have an additional source of evidence, the In-Silico one, why would you ethically or strategically accept to go with less than that and just remain with the current approaches? So the idea is essentially about trying to test earlier, to fail faster, and to also bring to preclinical testing similar situations or scenarios of testing in a virtual scenario that resemble as close as possible the ones that we're going to find eventually in the real world scenarios. And as Andrea and Daniel were indicating, there are situations where perhaps these models are not the most, this approach is not the best one, or where even if it is good, you will still want to have a confirmatory evidence through some sort of testing. But the question is, could we reduce the amount of testing to those studies that we are likely to be successful? And also, can we go to that situation where we put potentially people at risk in any study, or we harm animals? Can we do that in cases where we are as confident as we can be through modeling that this is likely to be a successful outcome? So that's what essentially In-Silico trials or In-Silico testing is trying to do. And behind that, the science of computational medicine, which is the use of engineering principles in the understanding of disease and the treatment of human diseases. Now, there is evidence out there that this has been, a successful approach, and that there are both returns on investments in terms of reduction of cost, but also faster time to market, and therefore bringing innovative therapies faster to benefit patients. The other thing is, one of the things that I wanted to emphasize is what is entailed into many of these techniques, and this is basically around the use of virtual populations and virtual patients. So how we build those, also how we develop mechanistic models, or models that are based on first principles from both diagnostics and interventional approaches, and then also how we try to, as much as is possible, capture aspects of human intervention, like, for instance, readers in the case of radiological systems, or measurements of outcomes that might be, also interacting with, for instance, with the lifestyle of a particular patient, or might be interacting with the way that particular clinicians approach the interventions. If you think about in surgical interventions, there could be different ways to do those, or different levels of skills. Now, one thing I wanted to also bring about is this notion of virtual trials, and to what extent this sounds similar, but it's actually different to in-silico trials. So to my understanding, most of the times when people talk about virtual trials, they mean fundamentally this umbrella term that focus on things like decentralized trials, remote trials, site agnostic trials, direct-to-participant trials, location variable trials, mobile trials. All of these different approaches are related more to the way we capture the human data, as opposed to the fact that we utilize virtual subjects. So these sort of approaches can be very good in terms of accelerating human trials, but essentially doesn't change the fact that you're still, putting people at risk. So when we talk about in-silico trials, and we talk about the need of access to real-world data, what we mean is literally real-world data from hospitals that can be utilized to actually build populations where the testing can be, from where we can build, say, anatomical models, physiological models of the interventions, and so on and so forth. So I'm going to give one example, and then I'm going to just skip to the last part, where I'm going to try to connect with some of the thoughts around regulations as well. So this is an example of work that we did in our lab that is close to what we do, where, for instance, in this case, it's a real-world device from a manufacturer. We built libraries of anatomical models based on radiological imaging. These patients, not all of them necessarily underwent a device. So in that sense, these are not patients that were part of any trial. They're patients that underwent imaging only. And from each of those, you can actually virtually deploy the particular stent and look at the influence of anatomy in the device response. You can also, based on wearable information or information from ICUs, you can test different scenarios of physiological regimes. So you can do essentially a virtual stress test or a rest exercise study. And you can also overcome the need to have randomization, because you can put multiple devices on the same subject and compare them head to head, which also circumvent some of the problems of comparability of multiple studies. And you can also develop certain surrogate endpoints that are the variables that we will sort of try to associate with risk or with success, with performance and with safety. And the trick of this is that you can do that at a scale, really at scale, by access to really large data sets, like population studies like UK Biobank, but also the richness of regional studies that we have with secure data environments, for instance, that the government is developing, or access to data like CPRD, for instance. Now, underpinning all of this, there are complex workflows which require new skills. And I'm not going to go into this, but we recognize there is going to be a need of new profiles that will have to have an interest in regulations, and also existing regulatory profiles that will need to be interested and be, develop expertise in additional methodologies as well. And I personally see that this as a great opportunity for more interdisciplinary and cross-sectorial collaboration. There's a number of examples that I'm going to just swap through without going into a lot of detail, but this is a study that I was talking about brain aneurysms. A typical pivotal trial is 20 to 40 million for each of these, and will take you about 8 years. We ran this in-silico trial in about 3 months, and we could actually get to 5% accuracy of correspondence with the other 3 trials that were in the literature. But more important, we could start looking at the effect, for instance, of hypertension. So these are normotensive subjects and hypertensive subjects. And if you do the whole population normotensive or the whole population hypertensive, you have already 15% difference in the results of that simulated study. Okay. FDA has been doing additional work in the area of imaging. So this is in-silico trials used for imaging systems, where you simulate the whole pipeline of acquisition and you compare it against the real one. And they publish, abundantly about this. Trials and the models and this that you use could be effectively, they are pieces of code, so they can be made publicly available to the extent that is appropriate, which means that there is a lot more transparency and reproducibility. Daniel, I think, was talking about preclinical studies. So this is a study by Zimmer Biomed where they show testing of devices and sort of stress studies where essentially replaces cadaveric scenarios in conditions which are much more thorough and much more exhaustive than the number of studies that you can do with cadaveric specimens. And then J&J, the PUI, has developed studies where in-silico studies where there is a combination of a device but also some sort of interventions. In this case, there is a wedge cut that you need to do on the bone and then put a fixation. So what you're doing in a trial is a combination of the device plus also the way that you do the surgical cut. And there's a number of documents that I can point you out where there is tons of similar studies delivered by FDA and other collaborators. So just to finalize, one of the things that comes over and over and over in this area, and I know is of important concern for our colleagues from MHRA, is the credibility of these models. So how credible they are. And there is frameworks that are emerging from standards bodies and from FDA itself, which develop risk-based frameworks where the amount of testing you need to do and validation and credibility factors, as they're called, or credibility activities, depends on the amount of influence that the model that we are testing will have in the final regulatory decision, but also in what is the decision consequence. So if we get it wrong, what is the worst thing that can happen? Is the patient going to die, or is it just a discomfort? So there are frameworks that we don't have time today to go in depth over those, but to actually do this and do it properly. And most of them start with the definition of a context of use, and there are multiple contexts of use for the same simulation platforms. That's why we need to be careful about how do we assess the clinical evidence that results from these simulation pipelines from the actual fundamental software that is being used to generate the analysis and the physics and so on. And there is work by FDA and others about trying to develop what is called medical device development tools, or MDDT, and drug development tools, DDT, which is regulatory approvals that are actually on the tools rather than on the medical product, so that you have some sort of certification of the tool itself. And one of the things that some of the colleagues in the CEiRSI we are discussing with the MPA, with the National Physics Laboratory and the CEiRSI as well, is do we need to consider a space for metrology laboratories, for instance, for doing part of the heavy lifting of the certification of the technical software and differentiate that from the actual more clinical testing that comes, that notified bodies, approved bodies, and MHRA will want to take responsibility for. So with that, I'd like to finalize and say that this is essentially what we are exploring further through the CEiRSI, and if people are interested in that, I'm very happy to expand. Thank you.
Alex, thank you so much. Do you know, whenever I sit in on your presentations, I'm absolutely overwhelmed by your dedication and enthusiasm to this subject, and we've come a hell of a long way, I think, in the last sort of few years in sort of progressing in-silico. So it's great that you can give us that update and those examples. And it's also really good that we can have folks like we have on the call this afternoon to really cement some of the thought processes around regulatory approvals as well. So thank you. I've got a couple of questions that have come through. We'll save those for the end. But Dominique, maybe I could introduce you. One area perhaps of the MHRA that I haven't come across actually, which is for scientific data and insight. So really interested to sort of understand how that fits. So Dominic, over to you.
Yeah, so actually there's not that much to add from my side here, and I will just very briefly say a few things, and it won't take long. So I promise this, which is a good thing, I guess, on Friday afternoon. So just so in terms of regulators and simulation, there has been quite a lot of interest recently, and there have been a number of meetings to discuss this and to find consensus on how simulation could be used in various regulatory purposes. I won't read this slide out here for you now, but you see this here. So we are not alone. I think this is what this means here. So, and also just continuing on from this, the simulation is, there are various levels of maturity from different government agencies, both UK and international, to what extent they're interested in simulation to base their decision and their information gathering and evidence generation on simulation. So there is the MHRA. So we certainly encourage simulation and modeling as part of new drug applications. Well, also others do this. So the FSA uses routinely simulations to predict toxicity in chemicals. But also DSDL, so the military in essence, heavily uses simulation and modeling for military to understand all sorts connected to things ranging from battlefield tactics to how to deal with supply chains or these sort of things. I guess most of the things we don't know because they won't tell us, but some of them we know. So when it comes to us, then one of the biggest barriers that that the system in a way has is that it's unclear what exactly is and is not acceptable in terms of evidence here. So in particular, so where we see simulation modeling as something useful is to plug evidence gaps. And simulation modeling is not meant to replace clinical trials, but to reduce uncertainty where clinical trials are not ethical or not possible, or other interventional studies are not possible. And the key here for the MHRA is that the validity of the approach when you have simulation as part of an application, that needs to be demonstrated credibly. And what precisely this means is, of course, sometimes difficult to know, but for this reason, the MHRA also encourages industry representatives to just approach the MHRA for scientific advice early, where these things can be discussed. So one of the one of the things that can become a problem, this is terminology. So one of the things that emerged is that often it's not quite clear what people mean by certain words. So the thing that's highlighted here, I'm not sure whether you can see my cursor here, is validation and verification is used in medical devices, whereas the EU uses the term qualification. So there's a need to just clarify what we mean when we use particular word. And also one of the items that's very important here is that the accuracy of any algorithm, so the analysis should be verifiable. So this comes back to the point I made in the previous slide about verification and validation, that this is something that's very, very important. Yeah, that was it. As I said, it would be very short because it's Friday afternoon. So if there are any questions, I'm happy to answer. Thank you.
Brilliant. Thank you very much, Dominic. And we're back on schedule. So absolutely fantastic. I do have a couple of questions. So maybe I can just get you guys to come back online with your cameras. But one or two. So question first of all for Andrea, if you're still there, or Dominique, if you want to pick this one up too. In the regulatory view, do you think that the virtual patients or in-silico trial based on PK/PD, which for a non-medicine person, I guess that's pharmacokinetic patient studies, right? Or QSP models, could QSP models replace or reduce the sample size, or at least added some evidence in clinical trials, especially and particularly in rare diseases? How do you see in-silico fitting in with things like rare diseases? Andrea first.
Well, thank you, Phil. It's a very interesting question, this one. So I think that in my presentation, we've seen this kind of diverse approach between the assessor in focusing on the clinical trial, so the quality, the non-clinical and clinical team, that they consider the in-silico as an ad hoc, as a supportive element. Meanwhile, the clinical pharmacologists, who are also computer modelists, so they do computer modeling simulation, they are more prone to support this new approach, if you like, as for the first in human. Because so far we tend to rely the vast majority of the time on animal modeling. And from the animal modeling, then we switch into a human being with the first in human trials. I think that the CEiRSI and phase 2 of the CEiRSI will provide a very strong clarity and piece of information regarding this question. As it stands, I think that we will tend more to look at support from In-Silico, not substitution. But going back to what Dominic was saying, the question of model reliability, what are the consequences of having the wrong model, for example? But what are the consequences of not having the model as well? So I think that probably what we are looking to see whether there are some standard or potentially a standardization process that will allow us to be more open, if you like, to this approach. But I think that the opportunity to work with the CEiRSI and the In-Silico team is great for us, because it will inform our decision-making process. It will open new avenues for us, and at the same time, potentially, we will be proud to consider to develop guidance that are fit for purpose for the In-Silico trial as well, of the new applications. I see the future very positive, but I know that we need to go through a kind of a stepwise approach. That's my personal opinion.
So actually, maybe I can pick up on that and bring in one of the other questions which has been asked. Thank you very much, first of all, for the question we've just had there. But just to pick up on another one from Jeff Bishkoff, who's on the line today, and taking that a little bit further, Jeff has asked whether where do you see the gaps existing between things like V&V and the FDA guidances and so on, and what do you think that you specifically need to be more to fully embrace In-Silico trials? And if I take that one step further and maybe include Alex in the conversation here, Andrea, I thought that was a really good point that you made, that you see In-Silico as a support rather than a substitution. So if, for example, the FDA are using their guidelines, and I'm not exactly sure how far they're using their guidelines, so please forgive my ignorance, but if they're actually using In-Silico as the basis for an approval, would that mean then, with this support idea in mind, that you might need additional information to actually get a product across that approval line? I don't know who wants to take that one on first. Andrea, you've got your...
Well, I can be the guinea pig, I don't mind. So I'm not familiar with VNV 40 guidance from FDA, to be absolutely honest. Of course, we use FDA and we use EMA as solid regulators. So we trust them and we tend to be aligned with their decision. But nevertheless, there are differences between us and them. I think that we have a great opportunity. We are a sovereign regulator, as they are sovereign regulator. And I think that, as I mentioned already, I think that we as regulator, we need to go through a stepwise approach. And the 9 project or 10 projects that are involved in the phase 2, the implementation phase, will put a lot of light, and they will help us to get a better picture and potentially improve the confidence as well. Because we tend to be scientists by default, and therefore we would like to see the evidence, and nothing is stopping us to change our idea if there is the ground for this to happen. Therefore, we are open to learn and open to collaborate. But I think that it would be difficult for us to adopt something that has been adopted by others, but that we've not been involved, and therefore we did not develop the confidence. Because what we need to remind ourselves is that we are conducting a regulatory review, therefore we're looking at patient safety. I'm not saying that in-silico trials are not safe, not at all. I am always thinking about the fact that having the in-silico modeling to support the trial, even when we move from the animal model to the first in human, is unbelievably good. It's something fantastic. And is also suggested by the clinical pharmacologist.
Brilliant. Thank you. Sorry, go. Yeah, sorry. Carry on.
No, and then I was saying, I think that what we need time to understand this. And it doesn't mean that it will be 5 years. Probably in a very short period of time, we will make our mind up. But I think that the most important part is to work together with Alex and the CEiRSI In-Silico team, where we can learn and exchange information. Then we'll build up our confidence.
Thank you. Dominique and Daniel, before I bring you into this conversation, Alex, maybe you could add some comments on the level of cooperation that you've had with the FDA on things like In-Silico. I mean, I know that you've had a lot of conversations. How has that figured in with the work that we're planning here?
Yes. So, sorry, and also as a heads up to Amin, who is here on the line, I mean, he could probably add some comments to the first question around the rare diseases, because he has quite some experience on those. But coming back to your question of the MHRA, sorry, of the FDA. So at the moment, as far as I understand, Phil, FDA doesn't accept as pivotal evidence in-silico, as far as I know. It may be that it does in some aspects of pharma, but at least in devices it doesn't. And it's more along the same direction that Dominic and Andrea and Daniel have been suggesting, that it's more as a adjuvant to the existing evidence. So I think the idea is that you go, as a company, you go with a lot more confidence to do your randomized control trial, and you save time and effort, and you avoid risk in the trial itself by utilizing this evidence. And I think in the foreseeable future, that's likely to be the focus. And then regarding the second part of your question, we know that there are certain special circumstances now with the participation of FDA colleagues, but they are technically part of the CEiRSI as well. And the idea is that in the case studies that we're going to be doing over the next few months, some of the teams have FDA involvement, and we hope they will be able to continue to collaborate with us. And because we think that actually the most important way, or a key way to build trust and confidence, but also harmonization globally, is through the regulatory science itself, right? Is agreeing on the principles, even if the actual procedures or pathways may be slightly different.
Thank you. I mean, before I bring you in on rare diseases, Dominic and Daniel, do you have any thoughts on that sort of cross-jurisdictional thought on how to accept or review data? Yes, Daniel, go ahead.
I suppose I would probably just echo the thoughts that Andrea put forward, but certainly in a cross-jurisdictional sort of approach, the MHRA does have dialogues with comparable regulators on various different subjects. So we have dialogues with the FDA, we have dialogues with TGA and others, IMDRF, etc. And these dialogues often cover various different topics, especially in around emerging regulation and areas of regulation. And I agree that harmonizing is beneficial for all, so that the expectations are clear globally makes it better for producers of devices. So they've got just one sort of global set of standards for this that is accepted everywhere, and they don't need to be aiming for different targets depending on the different jurisdiction. And so I think it's an area that could be picked up in terms of discussions with comparable regulators across the world about how expectations in these areas could be aligned.
Great. And that's the power of CEiRSI, right? In that, you know, we have a collection of minds that's sort of trying to drive an agenda. So that's fantastic. Thank you. I mean, let's bring you back in on the question of orphan drugs and orphan devices and CEiRSIs. How is that being, or how could that be managed?
Who? Sorry.
Sure, I can comment on that, but if you allow me, sort of, I just wanted, to go back to what Andrea, sort of talked about the comfort and Alex. I think Alex already knows that, I'm just coming back 1.5 hours ago. I arrived back from Washington, D.C. from my site visit to FDA. But the one of the things that was brought, obviously, and everybody agrees, is the level of the confidence, getting comfortable, sort of with, acceptance of these and balancing the, sort of any, let's say, sort of the harm, sort of that they may do by wrong, sort of simulations versus, sort of not having them. But one of the, I think, issues that we have got, because people, let's just be honest, there is a, sort of a huge gap between what FDA is there, and versus European side, I think, is the structure so far, and this is where CEiRSI can help hugely, is the structure, because it is in FDA, is centralized, while, sort of in the EMA side, with, sort of different groups, as well as, sort of the MHRA were part of that. This, we didn't have that. We started in my area, which is the PBPK, PKPD, and, sort of the connection with, the in vitro system, QSP, and so on. We started with both of these, organizations at the same time. Not many people will remember, but in 2005, we actually ran, a workshop in Berlin in the old B Farm place where the different regulators came. But at the same time that we ran, sort of in 2004 in FDA, but one of them continued on regular basis of engagement and workshops, and the other didn't. The reason it didn't, it was not that, people or assessors were not interested, but there wasn't a structure that allowed them annually to gather in place, for 3 days, 4 days, paid by their organizations, and go through the discussions and so on, and basically carry on with, how the things are changing, etc. And I think this is the biggest actually help that CEiRSI can actually do, which we have discussed with Alex, and that's the reason education part is actually a major part of it. So there is willingness there, but the structure, infrastructure has been missing, over the years, sort of for that. So if sort of I stop there, see if there is a reaction, we can move to, the orphan disease and how this has become actually one of the, sort of the areas that is getting much more traction.
Thank you so much. We've still got a few minutes left, so if you do have questions or follow-ups, please put them in the Q&A. One last question on here, however, though, is for you, Alex, and it's around some of the information that you showed on one of your slides, I think, which where you showed the number of medical devices, or the fraction of medical devices that don't actually progress to beyond a research stage. And Duncan is wondering whether or not you have similar information on the number of devices which don't reach market access, or if they do, where they don't produce a clinical benefit, and how we can use that sort of data to demonstrate the power or the the value of In-Silico. And his last question on that is, can you give any indication on what this fraction is and whether or not In-Silico trials could affect that proportion? Difficult question to answer on a Friday.
No, that's a fantastic question, actually, Duncan. And I'm glad that you asked it early enough. So I've been doing some search, because the short answer is no, I didn't know that fraction, or I don't know that fraction fully. So I've been doing a bit of search while the other questions were being answered. And I could only found one publication in the area of tissue engineered medical products that talks about a 7% being that progression. So clearly, I mean, if that is any reference about what is going on in other areas, is clearly a really good point, the one you're making, that you know, my estimate is still an optimistic one of what's going on, right? Which is quite depressing when you think about it. In terms of how you could overcome that. Sorry, and I'm going to be doing a bit of more search on that question, because it's really good. It's a really good point. How In-Silico could affect this? I suppose this depends on what is the main reason why these post-market failures happened, right? So just to give you two examples that are very different. If the post-market failure is due to the fact that your trials were not really representative of the population, then I think there is a hope that In-Silico methods might be useful there, because I am convinced that we can generate much more thoroughly representative large-scale virtual populations that we do at the moment trials. Sometime in my group, I say to the students and postdocs, you know, we should aim at having In-Silico trials which are 2 orders of magnitude larger than the equivalent trials for the same product that we're looking at in the current, you know, the current state of affairs, so to say. So if you're working in an area where the trials are 100 people, you know, we want to do 10,000. If we're talking about an area where is, you know, 1,000, we want 100,000 type of virtual subjects. And also make sure that extra patients are not just little variations around the ones we had in the first place, but they're actually spreading all over. And there are techniques to do that. And there is work going on in some of the colleagues of the CEiRSI. Now, there are other reasons why you may have failures post-market. One could be for you know, aspects of health economics, which probably means that you thought about health economics too late. But there is colleagues in around health economics in the CEiRSI that are starting to look at how we can use In-Silico models to estimate as well health economics well before we go to market. But there could be also other things that are more difficult to model, like, for instance, human factors, which, you know, learning curve. And some of those things, we have some ideas about how you may model them, but it's really, really very early times. So certainly a great question. And I think there is areas, I would say there is a potential for In-Silico, but not necessarily in every single thing that may be the cause of those post-market failures.
Thank you, Alex. And Duncan, I'm pretty sure that if you get in contact with Alex, he'll be more than willing to follow up on some of those answers. So we're running really close to the end of the session now. We've got about 4 minutes to go. So maybe what I will do, I'll use a little bit of chair's prerogative, I guess. I'm allowed to do that. So to you guys from MHRA, just a quick little round table, really. It's fantastic that you guys are on this call, and it's wonderful that Alex has managed to get you all together on such an esteemed panel, and it's just fabulous to have this conversation. But I'll ask the same question to all of you, and maybe you can give us some pointers as working on the CEiRSI. What do you think are the most important things for us as a CEiRSI to do next? What do you think we can do as a group which will help you as the MHRA become more confident in the outputs of the processes that we're talking about. So just a sort of an open question to finish the conversation so that we can maybe repeat this panel in a few months' time and see whereabouts and how we've progressed. So Daniel, maybe I could come to you first. What can we do next?
Sure, Phil. Well, I think by the looks of it, you guys and girls have done quite a lot. But I would probably just say that case studies, more case studies like the one that you have where, or the ones that you have where you've demonstrated that through modeling, you're able to get pretty much the same performance as past demonstrated performance in trials and in investigations, and across a breadth of different devices as well, not just for a single use case, but for a range of use cases, really would show the power of in-silico testing. And I think that that would be powerful evidence into how it could be accepted potentially as as supplementary evidence in future.
Brilliant, thank you. Andrea, same question.
Yeah, I think evidence is extremely important, but also keep working together, I think, is as important as the evidence, because the more we do together, the more we understand about each other, and the more we potentially converge in the same direction, which is patient safety, good medication, good devices, reduce the time to provide life-saving devices or medication to patient. And I think that yes, I agree. Two
Perfect.
ingredients: evidence and collaborations.
Brilliant. And Dominic, last word to you.
Yeah, I keep my video off because I had a little bit problems with the network connection. I think the main thing is to understand the concept robustness of models. So we talk about evidence and acceptability of evidence, but what's really important here is, so when we do modeling, simulation modeling, then that often can be very interesting modeling. You get very interesting results. But one of the questions that I think the CEiRSI would do well to answer is, what does it mean for this evidence to be robust? So I think Alex had this on his slides. All models are wrong, but some are useful, and usefulness is part of. So robustness is an important part of usefulness. And robustness here means that we understand exactly to what extent we can rely on it, and we know exactly how this slots in with other pieces of evidence. But we also aware of the assumptions that go in there, and we are very much aware of the uncertainties that are related to the results that simulation models produce. And then there are other ethical and so on questions connected to this. So, but precisely what does it mean for models to be robust? This is some in the context of this particular CEiRSI. That's something I think I would like to see to come out explicitly at the end.
Brilliant. Thank you so much. Well, before I hand back to Alex for a final word and before we close, I've really enjoyed the conversations today and the presentations. Thank you, first of all, for you guys at the MHRA to come online on a Friday afternoon. It really is appreciated. And I'm sure that our collaboration and cooperations are just going to continue and snowball from these sorts of conversations. So thank you so much for joining. And maybe then, as it's got to 4:45, and we've come to a finish, final word from Alex before we all head off for our gin and tonics.
So thanks a lot, Phil, for chairing the session, and also for our colleagues from MHRA. I was yesterday in a meeting in London. Andrea was there as well, where MHRA was presenting their experience in using AI techniques for improving operational efficiency and for a number of other activities. And I was impressed by the speed at which the agency is stepping up to new technologies. And so I'm really sort of encouraged by what I saw in that space, that their commitment to seriously consider techniques like in-silico technologies is strong. I would really appreciate it if everybody engages. So I saw Lara as well putting on the chat some of our mechanisms of communication and some of the upcoming events. All of these sessions will be recorded and released in the YouTube channel that Lara puts on the chat. So you will be able to also share them and follow us. And also a bit coming back to what Phil was saying, if there are specific topics that you would like us to explore in these webinars, let us know, because we are in a phase that we call now of awareness and training before we engage into a phase of case studies that we will do in a couple of months. And we want really to all of these events to be open and listen to and be responsive to what the community is needing as well. So with that, really thanks. And I want to just finalize by acknowledging our funders, MHRA as a partner that is supporting us, but also MRC and Innovate UK, who have provided general support for organizing all of these events. Thank you.